So this video is going to be about the second part of protein synthesis, translation. Before we start, just a reminder about two important molecules. First one is a ribosome, which we learned in Unit 1. This is made from both protein and rRNA, and it has both a large and a small subunit. We've then got tRNA, and two really important regions to tRNA. There's the anticodon and the three bases at the bottom of the tRNA molecule, because they're complementary base pair of the codon of mRNA, and you've got the amino acid binding site at the opposite end, which will bind to an activated amino acid and bring it to the ribosome. You do need to be able to compare and contrast tRNA with mRNA or with DNA. So you can see this has got a cloverleaf shape, whereas mRNA is linear and DNA would be sort of a linear sort of double helix. This is the shortest of the three, so it's the shorter than mRNA, shorter than DNA. It's an RNA molecule, so it contains uracil instead of thymine, just like mRNA, and it contains ribose as the sugar in the nucleotide, like mRNA, instead of deoxyribose. However, there is base pairing in tRNA. You can see at these regions here, complementary base pairing. There are some double-stranded regions, whereas mRNA is completely single-stranded. So before we start transcription, just a reminder, we've already had mRNA produced in transcription, exited through a nuclear pore, and travelling to a ribosome. That ribosome could be freely floating in the cytoplasm, or it could be attached to rough endoplasmic reticulum, which is usually found attached to the nuclear envelope. So once mRNA has reached the ribosome will bind, and two codons fit inside the ribosome. So here's the ribosome. Here are the first two triplets on mRNA, so codon 1, codon 2, and it'd be the start codon that the mRNA would begin at first, attached to first. You then got complementary base pairing, you can see the hydrogen bonds here, between the anticodon regions of the tRNA molecules and the codons on mRNA. That ensures that specific amino acids, which are represented by the circle and the triangle there, so two different amino acids being different shapes. So we've got specific tRNAs bring specific amino acids to the ribosome. A peptide bond then forms between the two amino acids. That requires energy from the hydrolysis of ATP. The ribosome then moves along one codon. So you can see the ribosome shifted along the mRNA by one codon. The tRNA molecule, the first tRNA molecule, has detached and left the ribosome, and that will go into the cytoplasm to collect another specific amino acid molecule. For the amino acid to attach to the tRNA, it requires the amino acid to be activated using energy from the hydrolysis of ATP. Within the ribosome, the process repeats again. Another specific tRNA molecule has come in. We've got complementary base pairing between the anticodon on tRNA and the codon on mRNA. And then you get a peptide bond formed between the adjacent amino acids, requiring energy from the hydrolysis of ATP. You can see as the ribosome moves along that the protein polypeptide chain starts to poke out this bit of the ribosome, and so does the mRNA here. This process continues with the ribosome moving along the mRNA, and the polypeptide chain is coming out of the ribosome. That will then start to fold into its tertiary structure, depending on the order of amino acids. So what might a long answer question look like? Translation mark schemes can change from one year to the next. This is kind of an amalgamated, so if you say all of this, you definitely get all the marks. And make sure you fully understand it if you can get all of these points in as well. So you can clearly see, so follow the steps on there. That should match what I mentioned in the video.